We can use the techniques of DNA technology to recombine and copy genes. A restriction enzyme is used to cut open a plasmid, a small circular DNA molecule obtained from a bacterium. The plasmid serves as a cloning vector. The restriction enzyme cuts only at a certain DNA base sequence, called a restriction site. The same enzyme is used to cut up DNA obtained from a human cell. The restriction enzyme produces DNA fragments that have matching sticky ends that join by complementary base pairing. An enzyme called DNA ligase forges covalent bonds that join human and plasmid DNA, creating recombinant DNA. The human gene is inserted in the middle of the plasmid LAC-Z gene. The plasmid also carries a gene for resistance to the antibiotic ampicillin. The human gene must be cloned to produce copies for use or study. A bacterium is induced to take up the recombinant plasmid from the surrounding solution in a process called transformation. This isn't as clear-cut as it sounds. Some bacteria do not take up the plasmid, and some take up plasmids whose LAC-Z genes lack the inserted human DNA. A bacterium is placed on a growth medium. It replicates the plasmid at the same time as its own DNA. Its descendants form a clone of bacteria that all have copies of the recombinant plasmid. The ampicillin in the medium limits growth only to plasmid-containing cells. But which cells have plasmids with a human gene? Bacteria without human genes have intact LAC-Z genes. This allows them to break down a substance in the medium that stains their colonies blue. Thus, the bacteria with human genes are in the colorless colonies.
All cells are able to synthesize ATP via the process of glycolysis. In many cells, if oxygen is not present, pyruvic acid is metabolized in a process called fermentation. By oxidizing the NADH produced in glycolysis, fermentation regenerates NAD+, which can take part in glycolysis once again to produce more ATP. The net energy gain in fermentation is 2 ATP molecules per molecule of glucose. Fermentation complements glycolysis and makes it possible for ATP to be continually produced in the absence of oxygen. 2 common types of fermentation are described here. Alcohol fermentation, which occurs in yeast, results in the production of ethanol and carbon dioxide. Lactic acid fermentation, which occurs in muscle, results in the production of lactate, lactic acid. Let's see the relationship between glycolysis and alcohol fermentation. Glycolysis produces NADH, ATP, and pyruvate, pyruvic acid. If oxygen is not present, NADH cannot be oxidized in the electron transport chain. Without fermentation, the cell would run out of NAD+, bringing glycolysis to a halt. In alcohol fermentation, the pyruvate, pyruvic acid from glycolysis, loses 1 carbon in the form of carbon dioxide, and the product is then reduced to ethanol by NADH. With the formation of ethanol, NADH is oxidized and becomes NAD+. With a continuous supply of NAD+, glycolysis can continue producing more ATP. During fermentation, the NADH produced by glycolysis is oxidized, ensuring a continuous supply of NAD+ for glycolysis. Alcohol fermentation occurs in yeast cells. Let's observe the relationship between glycolysis and lactic acid fermentation. Glycolysis produces NADH, ATP, and pyruvate, pyruvic acid. If oxygen is not present, NADH cannot be oxidized in the electron transport chain. Without fermentation, the cell would run out of NAD+, bringing glycolysis to a halt. In lactic acid fermentation, the pyruvate, pyruvic acid from glycolysis, is reduced to lactate, lactic acid, by NADH. With the formation of lactate, lactic acid, NADH is oxidized and becomes NAD+. With a continuous supply of NAD+, glycolysis can continue producing more ATP. During fermentation, the NADH produced by glycolysis is oxidized, ensuring a continuous supply of NAD+ for glycolysis. Lactic acid fermentation occurs in muscle cells.
The human immunodeficiency virus, or HIV, infects cells of the human immune system. Its primary target is a subset of lymphocytes called helper T cells, or CD4 plus T cells, so named because of the CD4 proteins that stud their surface. Proteins on the outer envelope of the virus recognize and bind to the CD4 protein and a co-receptor. The envelope protein is made up of two smaller proteins, GP120 and GP41. When the cellular and viral proteins bind, the cell membrane begins to fuse with the viral envelope. The viral envelope is left at the cell surface. This fusion allows the contents of the viral envelope, the viral core, to enter the cell. Cellular enzymes degrade the viral core proteins, leaving a few viral molecules in the cell cytoplasm, including the single-stranded RNA genome of the virus. Several viral enzymes, including reverse transcriptase and integrase, also remain in the cytoplasm. HIV is a retrovirus, which means that it uses RNA molecules rather than DNA as its genetic material. Using reverse transcriptase, it makes a DNA copy of the two single-stranded RNA molecules it carried into the cell. Reverse transcriptase also has two other enzymatic activities. With an RNase activity, it degrades the RNA. With a DNA polymerase activity, it makes a second strand of DNA. The resulting double-stranded DNA is transported into the cell nucleus through the nuclear pores and is inserted into the host's chromosomal DNA with the helper integrase. The viral DNA, now called a provirus, is about 10,000 base pairs long. The host RNA polymerase may transcribe the provirus at relatively low rates for several months or even years. However, if the T cell is activated by growth factors, cytokines, or antigens, the cell's transcriptional machinery increases the rate of transcription of viral DNA into messenger RNA. Some of the RNA molecules are processed, resulting in a variety of RNA transcripts. These RNA molecules enter the cytoplasm, where the cell's own ribosomes translate the mRNA into proteins. Some of the full-length mRNA transcripts will serve as new viral genomes. Ribosomes on the endoplasmic reticulum translate the RNA molecules that contain sequences for viral envelope proteins. These proteins are transported to the Golgi apparatus, where they are processed. The proteins then move to the plasma membrane and are embedded there. Free ribosomes translate other viral mRNAs. In the case of the structural proteins, a single long polypeptide is produced. Protease, another viral enzyme, cuts the polypeptide into distinct proteins. In this way, the cell's machinery produces many copies of both the viral proteins and the full-length viral mRNA molecules. Viral mRNA molecules and viral proteins, including reverse transcriptase, integrase, and structural proteins, are assembled into a viral particle that buds from the host cell membrane. As it buds, it takes a portion of the cell membrane and the embedded envelope proteins with it. A cell produces many viral particles during an infection, but ultimately the cell will die.
HIV reproductive cycle. Understanding the structure and life cycle of the human immunodeficiency virus, or HIV, the AIDS virus, is critically important to deal with the disease. HIV is covered by an envelope derived from a host cell membrane. Glycoproteins studying the envelope recognize and bind to receptor molecules on the host cell. A protein coat surrounds the viral genetic material, which consists of two molecules of single-stranded RNA. The enzyme reverse transcriptase enables the virus to make DNA from an RNA template, a trick for which this group of viruses is named retroviruses. The RNA molecules of HIV enter a host cell when the virus fuses with the plasma membrane, and the coat proteins are removed by enzymes. Reverse transcriptase catalyzes the synthesis of a DNA strand complementary to the viral RNA strand, and then a second DNA strand complementary to the first. The double-stranded DNA is incorporated as a provirus into the host cell's chromosomal DNA, where it may lie dormant for years. Occasionally, the provirus is transcribed into RNA. This RNA serves as both messenger RNA for the formation of HIV proteins and as genetic material for the next generation of viruses. Protein coats form around viral RNA and reverse transcriptase molecules. Viruses bud from the host cell, acquiring envelopes as they leave. Immune responses. The body's immune responses can be divided into the humoral, or antibody-mediated response, and the cell-mediated response. In both types of response, the helper T cell plays a central coordinating role. Microbes like bacteria or viruses carry antigen molecules on their surfaces that identify them as foreign. A macrophage is a phagocytic leukocyte that can recognize these antigens as foreign and engulf the microbe by phagocytosis. As a bacterium is broken down inside the macrophage, its antigens combine with class II MHC molecules and move to the surface of the macrophage. This class II MHC antigen complex then stimulates the next cell in the immune response. Helper cells interact with the phagocytic leukocytes to fight foreign bodies. The helper TH cell interacts with the antigen-bearing macrophage through its T cell receptor and the cell surface protein CD4. Each helper T cell receptor is unique, meaning that only a few helper T cell receptors can bind to the specific antigen class II MHC combination. The helper T cell is stimulated by this contact, as well as by the cytokine interleukin-1. The macrophage produces and secretes interleukin-1. Both interleukin-1 and contact with the class II MHC molecules stimulate helper T cells to grow and divide into many cells with the same specific receptors. Helper T cells next activate other cells in the immune system by secreting the cytokine interleukin-2. Helper T cells, cytotoxic T cells, and B cells are all stimulated by interleukin-2. Interleukin-2 causes helper T cells to divide more rapidly and increase cytokine production. Cytotoxic T cells are stimulated to become active killer cells. B cells are stimulated to develop into plasma cells. And then, in the nucleated cells of the body, bacterial, viral, or tumor antigens are gathered and presented on the surface by class I MHC proteins. These proteins are recognized by cytotoxic T cell receptors and the cytotoxic T cell surface protein CD8. Cytotoxic T cells are stimulated by contact and by interleukin-2 to release the protein perforin. Perforin molecules puncture the target cell, allowing water and ions to rush inside, causing swelling and lysis. In this way, cytotoxic T cells deprive pathogens of their host cells so they cannot reproduce. Cancer cells are also removed from the body in this way. The activation and killing action of cytotoxic T cells is known as the cell-mediated immune response. There are also B cells that are lymphocytes that produce antibody molecules in response to certain stimuli. Every B cell produces and presents its own uniquely shaped antibody receptors in its plasma membrane. These unique receptors only bind to antigens with specific three-dimensional shapes. The B cell that successfully binds to the antigen is stimulated to proliferate into a clone of cells with the same specificity. This process of antigen-selected proliferation is known as clonal selection. B cell proliferation produces clones of plasma cells and memory cells. Memory cells are long-lived cells that can respond rapidly upon subsequent exposure to the same antigen. They account for the rapidity of the secondary immune response and long-term immunity to a specific disease. Plasma cells produce and secrete antibodies in increasing amounts, reaching a peak 10 to 17 days after the activation of the original B cell. These antibodies flow through the body's fluids and tag foreign cells and molecules containing the original antigen for destruction. The production of antibodies for purposes of fighting an infection is known as the humoral immune response. The humoral or antibody-mediated response often requires the participation of helper T cells. Helper T cells are themselves first activated by cells which engulf the foreign cell. The macrophage digests the bacterium, freeing antigens that combine with class II MHC proteins. The macrophage now presents the class II MHC antigen complex to other cells of the immune system. Each helper T cell contains a uniquely shaped receptor. When a macrophage presents a specific antigen, only a few helper T cells in the body are therefore selected for activation. Let's see what happens to the B cell when it encounters the same antigen. A B cell binds to the antigens of a microbe and takes the microbe inside by endocytosis. After the microbe is digested, its antigens bind with class II MHC proteins and move to the cell surface. This B cell now is presenting the same antigen that fits in the receptor of the helper T cell. When the helper T cell receptor binds with the B cell class II MHC antigen complex, it begins secreting interleukin-2 and other cytokines that activate the B cells. Activated B cells divide repeatedly into memory B cells and plasma cells. The plasma cells produce and secrete large numbers of antibodies that are specific to the original antigen. In this humoral response to a T-dependent antigen, both the helper T cells and B cells are identified and selected for activity by the antigen. The binding of antibodies to antigens tags foreign cells and molecules for destruction by the immune system. Antibodies produced as a result of the humoral immune response effect destruction via neutralization, agglutination, precipitation, and complement fixation. Let's see how each of these mechanisms work. Neutralization of bacteria or viruses occurs when antibodies bind to and block the activity of the antigen. Neutralized viruses cannot invade host cells. Microbes and soluble antigens that are coated with antibodies are then eliminated by phagocytosis. In this process called opsonization, the bound antibodies enhance macrophage attachment to the microbes, allowing them to be easily engulfed and destroyed by phagocytosis. Agglutination is the process whereby bacteria or viruses are effectively neutralized by clumping. Antibodies have at least two binding sites, allowing them to cross-link microbes by binding to identical antigens on two different cells. Microbes that are tagged by antibodies are then eliminated by phagocytosis. In this process, called opsonization, the bound antibodies enhance macrophage attachment to the microbes, allowing them to be easily engulfed and destroyed by phagocytosis. Precipitation is the cross-linking of soluble antigens, molecules dissolved in body fluids, to form immobile precipitates. Soluble antigens that are coated with antibodies are eliminated by phagocytosis. In this process, called opsonization, the bound antibodies enhance macrophage attachment to the precipitates, allowing them to be easily engulfed and destroyed by phagocytosis. Complement fixation is the activation of a series of serum proteins, leading to lysis of a microbe. First, complement proteins bind to antibodies that have tagged a foreign cell. Complement proteins can lyse any cell that has been tagged by IgM or IgG antibodies. Some of the complement proteins are activated to form a membrane attack complex that creates a pore in the target cell. The pore formed by the membrane attack complex allows ions and water to rush into the cell, causing the cell to swell and lyse. Bacterial genetics. Bacterial cells can acquire genetic information from other bacterial cells in a variety of ways. Once genetic information is taken into a recipient cell, it can recombine with the recipient's chromosome and create an altered bacterial genotype. This tutorial explores the mechanisms of gene transfer in bacteria and the physical structure of the bacterial chromosome. Growth and plating. The main mechanism for transfer of genetic information in bacteria is conjugation. An elegant experiment in 1946 by Joshua Lederberg and Edward Tatum demonstrated this phenomenon. In this experiment, two auxotrophic strains of Escherichia coli with different nutritional requirements were studied. One strain lacked the ability to make methionine and biotin, MET-negative, bio-negative. The other lacked the ability to make threonine, leucine, and thiamine, THR-negative, LEU-negative, THI-negative. First, the two strains were grown separately in liquid culture medium that was supplemented with all the required nutrients for each mutant strain to survive and grow. Each strain was then separately plated on minimal medium. This medium lacked the nutrients required by the auxotrophs: methionine, biotin, threonine, leucine, and thiamine. No growth was observed on the plates, meaning that none of the cells from either of the cultures had spontaneously reverted back to normal cells, prototrophs, capable of making these nutrients. Mixing two auxotrophic strains. Next, the cells from the two strains were mixed, grown together for several generations, and then plated on minimal medium. Only wild-type bacteria, prototrophs, are able to grow on minimal medium. Because the number of prototrophs recovered was too large to be accounted for by spontaneous mutation back to the normal genotype, the prototrophs must have arisen as a product of genetic exchange or recombination. Genetic recombination results in a wild-type strain of cells, MET-positive, bio-positive, THRU-positive, LEU-positive, THI-positive, able to grow on minimal medium. These wild-type cells occur at a frequency of 1 over 10 to the 7th power of total cells. The F factor. Lederberg and Tatum concluded from their experiments that genetic exchange had occurred between two strains of bacteria. Later, it became clear that this exchange, called conjugation, is unidirectional and requires physical contact between different strains. The different strains are designated F plus and F minus, with F standing for fertility factor or F factor. F plus strains are donors of chromosomal material, and F minus strains are receivers. The F factor is a mobile element that confers the ability to a bacterium to donate part of its chromosome. It is a plasmid that directs the synthesis of pili, tube-like extensions of the cell that allow contact with other cells. During conjugation, the two DNA strands of the F factor separate, and one strand moves into the recipient cell. Both strands serve as templates for DNA replication, resulting in two F+ cells. The F factor itself is passed to all recipient cells, but on rare occasions the transfer of genes from the bacterial chromosome, genetic recombination, also occurs. Evidence for conjugation requiring physical contact was provided by Bernard Davis's experiment, in which F+ and F- strains that were auxotrophic for one or several amino acids were grown in opposite halves of a U-tube. The halves were separated by a filter with pores large enough to allow the medium to flow, but small enough to restrict the passage of bacteria. After incubation, Davis plated bacteria from the two halves of the U-tube on minimal medium. He found that no bacteria grew on these plates. Therefore, physical contact is required for genetic exchange to occur between the two strains. HFR strains. Cavalli-Sforza and Hayes have described strains of bacteria that undergo recombination at a high rate in mutagenesis experiments. These strains are called HFR, high-frequency recombination. HFR strains result from integration of the F factor into the bacterial chromosome of an F+ strain. Chromosome transfer. The arrangement of the genes on the bacterial chromosome can be determined by using interrupted mating with different HFR strains. In this technique, during conjugation between an HFR cell and an F- cell, the mating is interrupted mechanically at specific time intervals before the entire chromosome is transferred. This allows the gene order and the distance between genes to be determined. Chromosome mapping. Evidence for the circularity of the bacterial chromosome came from using different HFR bacterial strains. Each strain gives different HFR linkage maps. During conjugation between HFR and F- cells, the position of the F factor determines the initial point of transfer. The order of gene transfer in 4 HFR strains, as given in the table here, suggests that the E. coli chromosome is circular. The point where transfer originates, the origin, or O, see red O in diagrams, is determined by the point of integration of the F factor into the chromosome. Transfer can proceed in either direction, depending on the orientation of the F factor in the bacterial chromosome. HFR reversion to F+. The F factor can lose its integrated state, causing an HFR strain to revert to F+. In this case, the F factor may carry adjacent bacterial genes along with it, leading to an F' strain. An F' bacterium can initiate conjugation with F- cells and may transfer chromosomal genes to the F- cell on the F factor. In this case, a partially diploid cell, a merozygote, is formed. The recipient cell has a complete chromosome plus the newly introduced chromosomal genes. Transformation. Transformation is another method of transferring DNA in bacteria. It provides a mechanism for the recombination of genetic information in bacteria. Cells must be competent, that is, able to take up DNA. Double-stranded DNA enters the bacterial cell, and one of the strands is degraded by nucleases, whereas the other strand integrates into the bacterial DNA. Conjugation, the main mechanism for genetic exchange in bacteria, is a unidirectional process that requires physical contact between F+ and F- strains of bacteria. F+ cells contain the F factor, an extra chromosomal plasmid that directs the synthesis of pili through which DNA is transferred to an F- cell. HFR, high-frequency recombination strains, undergo recombination at a high rate due to integration of the F factor into the chromosome of an F+ cell. They are particularly useful for determining the arrangement of genes on the bacterial chromosomes. HFR strains can revert to F+ strains if the F factor is excised from the chromosome. But an F' strain results if part of the bacterial chromosome is excised with the F factor. Thus, genes that were once part of the F+ cell's chromosome may be transferred to an F- cell during a subsequent conjugation, causing the F- cell to become a partially diploid merozygote.